Hi, in this video we're going to continue with single-cell RNA-seq data analysis. And the next step in 10x single-cell data processing is dimensionality reduction. For that we have a dedicated block which has the same name, and we're going to add it to the project. Now the idea behind this step is that initially we have a very high number of dimensions. We have thousands of cells, and each cell has thousands of genes that are expressed. It is hard to identify trends when we have so many dimensions. So dimensionality reduction is a method which uses principal component analysis as well as other algorithms to identify the most important dimensions in your data to preserve biological variance while reducing the dimensions to a much lower number. Essentially, we do not need the expression of, let's say, 20,000 genes for every cell to identify what type of cell it is. We just need to understand which genes are detrimental for a certain cell. So here, the only thing that we need to select is just our dataset. Now we also have an option to select a batch correction metadata column. Right now, if I click here, I will just see the name of the column from my metadata. This is the metadata I added for my samples, and I'm not gonna select anything here because genotype in this case does not represent any batch. But for instance, if you have data in your data set that was sequenced in different runs, for example, you have 20 samples in your data set and 10 samples were sequenced in run 1, then 5 samples were sequenced in run 2, and another 5 were sequenced in run 3. And you can add this information about which run each sample comes from to the metadata, and then here you will see this column, whatever you call it, like for example a run number. And then you would want to select it so the software will automatically correct for differences that are determined by the batches. Because for example, if you randomly distributed your samples among different runs of sequencing, you do not expect that the patterns of gene expression will follow the distribution of the runs. So in this case, I'm not going to select anything, but according to your data, you might want to change that. So now I'm just going to click run, and this block will generate a UMAP, which you can see right here. These are our samples, and this is a two-dimensional structure representing gene expression normalized by the reduced dimensions. And we also have a t-SNE plot, which is another common way of looking onto single cell gene expression. Now, if you would have selected and added a batch correction for each plot, you would also see a corrected version. So you will have a UMAP and UMAP corrected, implying batch correction, and also t-SNE plot and t-SNE plot corrected for the corrected version. Now, that is all you need to do in this block, and now you can proceed to downstream analysis.